My name is Martin Poulet. I'm a senior lecturer in the Department of Haematology at the UCL Cancer Institute. So cancer therapy is undergoing a radical transformation. We are testing a whole new way of treating cancer with engineered T cells. These T cells can be engineered to recognize cancer through a chimeric antigen receptor, and after administration they can engraft inside patients, proliferate, extravasate, home to disease, and persist for weeks, months, or even years. This form of treatment is showing tremendous promise in patients with disease that has been refractory to standard therapies. There is, however, a very pressing need to be able to track these cells in patients after they've been administered, which is a particularly challenging problem since these cells can persist for such long periods of time. And this project involves combining our experience with engineering T cells and with the experience in CABI in making molecular reporters to be able to track these cells over long periods of time. So Tammy is going to explain to you how we can additionally engineer these cells to be selectively trackable using PET and SPECT tracers in living human subjects with strategies that will allow us to track these cells over many weeks, months, or even years.
My name is Dr. Tammy Kelber. I'm a research associate and I work at the Centre for Advanced Biomedical Imaging, where I develop new imaging techniques to look at tracking cells throughout the whole body. So in the process of cell tracking, we utilise genes and insert them into cells. These genes then can either uptake radiotracers for nuclear imaging such as SPECT or by the firefly gene that produces light for bioluminescence for optical imaging. We can then light up cells and track them in 3D throughout the whole body. We can combine this with high-resolution MRI and use this to evaluate tumour volume and also therapeutic effect, corresponding cell uptake with therapeutic outcome. The collaboration between Martin Poulet's group and the Centre for Advanced Biomedical Imaging provides not only T-cell therapy expertise and genetic expertise, but also 11 state-of-the-art preclinical imaging systems and a highly dedicated cell imaging team. This is obviously a highly unique environment for not just only the development of molecular imaging techniques, but also the translation of T-cell therapy to the clinic, and puts UCL at the forefront for these techniques. There are only 2 research centres in the whole of the world that can carry out this type of research, the Centre of Advanced Biomedical Imaging being one of them. This is therefore a fantastic opportunity for someone to come and do a research project at a world-leading institution.